All right, we are going to get started, everyone. So I don't know how many of you were here a week ago, but this is part 2 of the lectures on variants and genes and their interpretation. So I'm going to, today's talk is on interpretation of sequence variant pathogenicity. And so I did want to just start with just broadly thinking about what are the types of interpretable variants that are found in a genome. And I sort of put them in roughly these 5 categories. So variants that cause monogenic diseases, where you may find them in the carrier state for recessive or actually putting you at risk as a dominant disease. Small effect risk alleles for complex traits, generally identified through GWAS studies. Pharmacogenomic variants that predict drug response or the risk for adverse events. Blood antigen variants that are important in thinking about transfusion risks, benefit for donation as a blood donor, and variation that's used in determining ancestry, which of course can roughly be any variation. So those are the kinds of variants that we may know and be well established. Most of what we struggle with are the novel and very rare variants where we have limited information for monogenic disease risk. And that's really what we're going to talk about today, is how we go about thinking about the evidence for variants and how to interpret them. Oops, sorry, is it going? And you know, in some cases, we'll be able to infer effect by looking at predicted loss of function variants like frame shift, nonsense, canonical splice effects. In other cases, not really obvious what the effect is, and you go about searching databases and literature for observations and reported effects and aggregating that evidence to try to classify a variant. Did that work? Awesome, thank you. It's inhibiting my ability to walk around when I don't have a remote. So this is largely what we're going to focus on, is how we go about doing this. And I do want to point out one really important fact, that most of the variation that we detect when we're doing monogenic disease testing, and this is for disorders that are largely Mendelian, but they span things that even are more common, like hearing loss and eye disease, hereditary cancer, hereditary cardiovascular disease like cardiomyopathy, arrhythmogenic disorders, etc. So there's a variety of moderately common rare diseases, as well as the very rare orphan diseases. And when we do testing like that, and this is just a figure from a paper where we looked at results from 15,000 probands for monogenic disease testing, and showed that the vast majority of variants, and there's a hash mark, this goes 3 floors up, we find only once. We get one shot at interpreting that variant, particularly if you're only looking in your own lab's data. Handfuls of variants we see more often, but most of that variation is extraordinarily rare and unique to families, and we have to do our best interpreting that variant. And not surprisingly, when we started a project to get laboratories, particularly clinical laboratories, but also researchers, to share their interpretations of variants, which we now do in ClinVar, we identified that of the variants where there were more than one submitter, 17% of those variants were interpreted differently between laboratories. So that was identifying a huge challenge and problem in the healthcare setting. And in fact, we have our first lawsuit still going on right now, where a laboratory testing company, in this case Quest, who owns Athena, was sued by a family who claimed their child died because of a misclassification of a variant. In this case, it was actually the lab called it a VUS, and the family thought it should have been called pathogenic, and if that had been known, the child would have been not treated with a particular drug that they claimed led to the child's death. So, making these decisions about VUS versus pathogenic have incredibly important consequences for the health and care of many patients. So a few years ago, a group of us at the American College of Medical Genetics and the Association for Molecular Pathology gathered together to try to give better guidance to laboratories on how to interpret variants, recognizing that we're dealing with very limited pieces of data in many cases. We don't have, like in the prior talk, 100,000 people with the variant or even 100 people with the variant to get details from. So we spent actually 2 years trying to agree on a set of rules to try and interpret variance. I love to show this picture where we're all drinking wine. It was not celebrating the completion, but the start of the process, we realized that we could only ever agree if we had a lot of alcohol involved, which is a joke, but to some extent, it was a very big challenge to actually agree on what constitutes very strong or moderate or other levels of evidence, because a lot of this evidence is not quantitative and not able to be used in a statistical manner, and it is left to professional judgment. That makes it very hard. I should clarify that when we talk about variant interpretation, I think about 3 stages of looking at the effect of a variant. One is, can you figure out if it's disrupting the gene? But what we really focus on, particularly in the clinical lab, is does that impact actually lead to a disease phenotype? That is pathogenicity assessment. And one of the aspects of the guideline that we wrote was actually to define 5 terms that we would use as a community to define different degrees of pathogenicity and confidence. So pathogenic, likely pathogenic, uncertain significance, likely benign, and benign were the 5 terms that were largely being used by the majority of labs, and we sort of solidified those. But that assessment of pathogenicity, which is can this variant cause disease in an individual, is separate than what we often do in a clinical lab, which is then to say, does this variant, with my interpretation, actually cause the symptoms in my patient? And that's a separate question. You go to a pathogenic variant, but say, I don't think it's causing this observed phenotype that I'm being asked to interpret. And in those cases, we use terms like positive, negative, inconclusive, carrier, things like that when we're saying, did any of these variants I find actually explain what I'm being asked to interpret? So we're going to focus on number 2 here. How do we assess pathogenicity? Does this variant cause disease? I pointed out we used the term likely here. We actually debated a lot of, what term to use when we were trying to say, well, we think this does this, but we're not fully sure. And we actually looked up, is there a quantitative definition for the word likely? Of course, there is not. And we actually surveyed laboratories and geneticists signing out cases. What's your definition of likely when you use this for variant assessment? And we got everything from 50% to 99% in this survey. So we said, okay, we need to actually just, for our community, for this purpose, define the word likely. And by popular vote, we determined that likely would be 90%. Why did we come up with that? Largely around what we felt clinicians would find it useful in terms of making clinical decisions. I'm feeling 90% confident in this interpretation. Is that enough to go down a path of treatment? And that was sort of subjectively how we thought about the utility of that word and the utility of that classification. Now, I should say that most of the variants we classify into the category of likely pathogenic is largely not based on coming up with a numerical endpoint that says, oh, this is 89% and therefore it's below the fold, or this is 91%. Almost never do we actually have a multi-likelihood, ratio or percentage that we can actually say, yes, this is 90 whatever. But we're really trying to say, based on your assessment of the evidence, do you feel 90% confident in the assertion you were making? That's what we're trying to arrive at, or what we sort of settled on to deal with this field. And so we then, based on aggregating lots of evidence approaches from many labs, we listed, I forget how many are here, but this many different pieces of evidence or types of evidence that we use day to day for asserting and interpreting pathogenicity. And then we segregated them by level, very strong, moderate, and supporting. We did the same for criteria we use to call things benign, and 3 levels. Standalone, that's really just the frequency is so high it is impossible to cause this disorder with a prevalence of X and a penetrance of Y. Or strong or supporting. And so we're going to go through, probably don't have time to go through all of these pieces of evidence, but we're going to go through some to kind of get a sense of the kinds of evidence we're using and how we're using it in different ways to interpret variants. So this, I made this sort of rainbow colored grid to make it a quick cheat sheet. Of course, what I didn't know is that people would take this cheat sheet, not read any of the paper, and try to use it. That's not recommended. There's a lot of detail in this guideline, and it's important for using these different codes of different strengths and different types. And then we came up with a bit of a sort of menu of how you combine different pieces of evidence with these different strengths to arrive at the pathogenic, likely path, benign, likely benign, and uncertain categories, which probably, as you'll see later, could be subject to some rearrangement. I'm going to go throughout the next 45 minutes or so a particular variant that I can illustrate a number of these pieces of evidence in a real context of interpreting a variant to help you sort of understand how we're doing this. This particular variant was one of the variants used in a paper we published from the CSER Consortium, Clinical Sequencing Exploratory Research, an NIH-funded program of introducing genomics into the practice of medicine. And we had 6 of the labs that are part of the CSER Consortium interpret a whole bunch of variants and then look at concordance and also look at the ability to resolve discrepancies by communication and sharing evidence. And so I'm going to show you the process that we went through for this one variant, which we then did for many variants, to look at concordance. So this particular variant is in the gene FBN1. That gene encodes fibrillin. It is the gene associated with Marfan syndrome, which is a syndrome involving skeletal, ocular, and cardiovascular phenotypes. The mutation spectrum is something you always want to define and know before you go about trying to interpret a variant. Is this a mutation or a gene for which the only types of variants that cause disease are missense gain of function, and loss of function don't cause disease? You need to know that so that if you see a loss of function, you don't say, oh, that must be pathogenic because it disrupts the protein. No, some diseases are caused only by the presence of protein gaining a function and disrupting other wild-type activity. So you have to know the mutation spectrum. In this case, all types of variation have been seen to cause disease. Missense, in-frame tend to be more severe, actually, in this case, than null, but both are pathogenic. There's extensive intrafamilial and interfamilial clinical variability. Patients in the same family with the same mutation can have very different phenotypes, but the penetrance, whether you have any evidence of disease, is quite high for this disorder. And nearly all Marfan syndrome patients have an affected parent, or it's a de novo mutation, again, related to the high penetrance. There are other phenotypes that are seen with varying evidence for those gene disease implications. So this particular variant, missense variant, was interpreted by 6 labs, and you can see how each of the 6 labs interpreted it. 3 said VUS. 2 of the labs had 2 different people in their own lab interpreting it, and in some cases didn't agree. So we had likely path and VUS from 2 different people, likely path from another lab, and path and likely path from 2 different people. So full spectrum of different interpretations for this variant. So I'm going to go through the Acmg framework looking at the different types of evidence that were brought together for this particular variant. So the first type of data we very often use is population data. How often is it seen in cases, and how often is it seen in controls? So each of the labs, I don't know where the 6th one went, but had to go out to the literature and find any reported patients with disease that were out there. Now you can see in this column, they all have a different number. 11, 3, 10, 7, or 9, which was noted to say, I read several papers and I couldn't determine if the patients in those papers were the same ones because there was overlapping authorship. That's a huge issue. I remember one case, a fellow came to me with the evidence for a clinical case on Usher syndrome and said, this variant's been observed 4 times in the literature. And I said, okay, and I looked into it and ultimately saw that there was overlapping authorship in all 4 papers. And in the end, we were able to discern that it was one patient in all 4 papers. And in fact, the clinical patient we were testing was the patient who'd never gotten his results back. There was one patient in the world. So this is where you can go awry. So sometimes you don't know how many patients there are because you can't figure out across papers if they're actually unique individuals. Some of the groups used at that time, we were using ESP, so the numbers of controls that often are used, or ESP, were looked at. Some calculated an odds ratio and did or a p-value on the case versus controls. Some used controls from locus-specific databases, et cetera. But this gives you a sense of how people come up with different answers to cases. The other problem here is that the case data actually came from varying phenotypes. Some had a diagnosis of Marfan syndrome, some were Marfanoid, which has very subtle features. It may or may not be Marfan syndrome. Some had a disease, Sprinson-Goldberg syndrome, that was later refuted as an association with FBN1. So, just counting up cases reported in the literature is dangerous if you don't look at the detailed phenotype and ensure they actually have the disease. So there was a variety of different pieces of evidence. Some said there was a statistically significant increase, but as we all looked at this data and the limitations, we said at best it is not fully statistically significant given the weaknesses. At best, we could call this a moderate piece of evidence based on the observations in these cases. The next is computational predictive analyses. People love to use these because they're easy to access tools. We asked labs to track which tools they were using. And so the teal shows which tools each group used. You can see there's like zero overlap in who's using what tool. And completely discordant results across each of these tools. All the labs said this does not meet the criteria to which we defined as all computational algorithms used indicate the variant has an effect. So largely this was not useful. One lab did try to conclude that there's another variant at the same site that invoked this rule that another pathogenic amino acid is at the same site. However, if you actually look at this variant in the literature, one case, no segregation, no other evidence, that variant cannot be said to be pathogenic. And so you can't then use it to implicate that position in the protein. So that was rejected. The next question, so none of the evidence up there in that second row was counted. Looking at functional data, we have this rule, located a mutational hotspot or known functional domain. If you go into the full description of this, you really have to find a region that is a small, incredibly well-studied and defined region, like the pore domain of a channel protein or something, that is fully devoid of any benign variation, very well studied. And if your mutation is in that region that's well known to just never tolerate pathogenic or benign variation, then you could use that as a piece of evidence. But in this case, if you go into any of the protein databases, you'll see some label on pieces of proteins like this, calcium binding conserved EGF domain. That does not mean that therefore anything in there is pathogenic. So one group decided that because of this, it was considered moderate evidence. I would argue that through our consensus, we all agreed that that was not moderate evidence. You can look at protein structure and the location of variation. So this is the PRKG2 gene. If you look at location of variance, everything out here is always benign. All the pathogenic variants sit across the CBS2, domains. And so that can help guide you in thinking about whether there's a likelihood it will be pathogenic. But you really have to look carefully. And if you look across the exact database and all the brown indicates just population variation, you will see a higher density of benign or population variation here compared to these domains. But there's still variation out there. So you can't just say, oh, all of this over here is pathogenic. And one group that's really done a good job at very specifically annotating which residues, when disrupted, are sufficient to say that there will be an impact, is the Enigma Consortium that does interpretation of breast cancer genes, BRCA1 and 2. And they've gone through each of the domains and very specifically said residues 1 to 101 are critical, and 81 to 99 and so on. They document these two regions, they would then interpret as pathogenic if you affect these residues or those, but not all of these would be uncertain. They use class 3 for uncertain. So there is, guidance that can be developed to specifically guide location in a protein as to its potential likely effect based on other studies of pathogenic variation. Next in this Marfan variant, looking at segregation data. In this particular case, there was no good segregation studies. One lab had clinical testing from one family. The proband was a 3-year-old with a diagnosis of Marfan, and later, the father came in and he was genotype positive, phenotype negative. Now, I told you earlier that Marfan syndrome has very high penetrance, but it's not 100% penetrant. So genotype positive, phenotype negative is often very difficult to use as evidence unless you are absolutely sure the disease is 100% penetrant. And the age of the individual correlates with the age of penetrance. So this rule for segregation, non-segregation with disease, is largely talking about phenotype positive, genotype negative. You know, when you see an affected individual and you're looking at a variant and they don't have it, that's pretty good evidence that that variant is probably not causative, assuming you've got a disorder without a high phenocopy rate, right? So that people might just have the disease for other reasons. And I study hearing loss. I can tell you a lot of people have hearing loss for reasons besides genetics. So not segregating in a family with an individual with hearing loss, more difficult to apply this rule. But a syndrome like Marfan syndrome has very discrete phenotypic features. You could largely say if they don't have the variant, that variant is probably not causative disease. So in this case, you can't really apply this rule at a level of strong because it was the opposite, genotype positive, phenotype negative. However, you could say, given that Marfan is fairly high penetrance, whoops, thought I had, so you could, we ended up arguing that it was supporting evidence a little bit. I think I may have had that slide in a bit. I think I was just describing segregation in terms of that it should, you know, the phenotype should always have a positive. So here's where we sort of said, all right, you know, this data argues against pathogenicity, but there is some incomplete penetrance, so we scored it as supporting evidence, not strong, for a benign impact. In the guideline, the guideline does not give specific guidance over how to score quantitatively segregation data. I actually fought for like 6 months to get the committee to put quantitation in this, and I lost that battle. I at least got us to draw a line that said more data was stronger. But now ClinGen, our ClinGen group is working to quantitatively define this. In fact, Stephen Harrison, who's in the room here, and Les Biesecker co-chair our sequence variant interpretation work group for ClinGen, which is sort of building more guidance and specificity and details into the ACMG guideline. And some of the different rules that we're working on are listed here. That this group also reviews all of our disease-specific expert panels in ClinGen that are coming up with very specific sets of rules for individual diseases and genes to give more specific guidance, like, you know, a functional assay. Well, what functional assay in what system does that count as strong evidence? Things like that. So this group approves and those sets of rules or gives feedback on. So that's sort of how that works. And one of the things that we've done is structure how you code. You use a rule, but you change its strength to code it in that way so that we can machine gather all of these codes that are being applied to different variants. So we've gotten a few of our expert panels, and there's actually a publication from Gail Jarvik proposing quantitative guidance around use of segregation within the ACMG guidelines in terms of number of segregations for a dominant family, equating that to a LOD score, and then applying it, supporting moderate and strong. So, you know, they're not all in sync yet, and it may vary how you use segregation in terms of how many phenocopies may be present for that given disease. And for our MYH7 group studying cardiomyopathy, they applied it a little more stringently on the supporting side than our RASopathy group, because there is slightly more phenocopies for cardiomyopathy than the RASopathy disorders, for example. So we are still in the process of coming up with the quantitative guidance through the ClinGen effort. We've actually come up with tables to help clinical labs count up affected and unaffected and arrive at a LOD score without having to calculate it, and then plug it into these formulas. So counting segregation is important, and that's actually a quantitative piece of evidence. However, it's important to remember that segregation of a particular variant with a phenotype is evidence for linkage to the locus, but not evidence of the pathogenicity of the individual variant, right? you could have that variant be in linkage disequilibrium with a different variant that's actually the cause of one. The LOD score will still be very, very good with large families, but in fact, it could be a different variant that's causing the impact. And so in the ClinGen group, we are working on not allowing segregation data to lead to a pathogenic classification unless there is specific type of data that definitively or that implicates the variant itself, like de novo occurrence or functional study, things that are really specifically saying, no, this variant is the one that's causing the effect. And observing a de novo variant at that position is good variant-specific evidence, whereas linkage is really locus-level evidence, right? So we can say, okay, if you're using this at a strong level, then you have to use some of these to make sure your totality of evidence that gets to pathogenic ensures it's the specific variant impacted. So that's kind of how we're working on that. It's still kind of in development. If you go back to our FBN1 variant, there was no de novo occurrence in this particular case. And I will say, and we talked about this last week in the gene level evidence, where once you've got a phenotype and you already know that gene is strongly associated to that disease, then the observation of a de novo variant is actually very good evidence because you've narrowed down the space by a small, either one gene associated with that phenotype or maybe a handful, where the statistical likelihood that you observe a de novo variant in that small set of genes or one gene is very, very rare. Whereas when we do exome analysis looking for novel genes, the statistical likelihood you will find a de novo variant across the coding exome is roughly 100%, right? So very different statistics, and that is why we use the evidence in very different ways for interpreting variation in a known gene already with an established phenotype association versus just looking randomly across the genome around prior probability. So that's sort of what this slide says I showed last time. We also have some rules that center around allelic data. So for example, in a recessive disorder, if we have a pathogenic variant in one copy of the gene and you have a patient with a phenotype matching that gene, variation on the other allele is actually statistically more likely to be pathogenic because of the sort of guilt by association. So we consider an observation like that moderate evidence towards pathogenicity if the variant is detected in trans on the other allele with a known pathogenic variant. Likewise, you can look at support for a benign effect if you observe a variant in trans with a dominant variant, particularly if the disorder is embryonic lethal with two impacts, right? So you can, say, well, if the patient, and in fact, this is how the Gary Cuttings studies around cystic fibrosis variation were done. They used the fathers of a proband with CF who carried one of the pathogenic variants and gave it to his child. And it is well established that cystic fibrosis men cannot reproduce if you have two copies because of the effect of the vas deferens and the inability to reproduce. So they could look at all variation on the other allele in a carrier father and rule all variation out as pathogenic if he had a biological child. Very smart. So that kind of, it varies by disease how much a variant on the other allele, it can be interpreted that strongly. But nonetheless, it is supporting, and for different diseases, you could decide that that's strong or even definitive, like in the story of CF. Also, if it's observed in cis with a pathogenic variant, less likely to be pathogenic. So these kinds of allelic observations can be used as evidence. And here's, doing cis-trans testing is very important. Here's a patient who has a pathogenic variant on one copy of the connexin 26 gene, and then you find a variant on a second variant, but you don't know cis-trans. Can't really do much in terms of interpretation. But if we show that the variant on the other was in cis, it is much more likely to be benign. On the other hand, if we show each parent is a carrier and these variants are in trans, it does not prove pathogenicity, and we would leave it as a VUS. So that's sort of when we show that it's in cis, it's much more likely to be benign, and we can use that to help rule out or demote variants. We are trying to come up with slightly more quantitative, in this case semi-quantitative approaches for, well, what if you see multiple observations of intrans? And a lot of the variants, particularly for recessive diseases, the only evidence we ever generate are intrans observations. So we started to come up with a rubric to say, how many points do you get for each proband, and whether you know that it's in trans or the phase is unknown, and whether it's homozygous versus compound heterozygous. I'm not going to go through all of this, but trying to give more guidance as to how to apply the PM3 rule with different levels of strength based on the numbers and types of observations of in-trans occurrence. So getting a little more sophisticated. This is our hearing loss group that's doing this. There's a few other pieces of data, like the patient's phenotype is highly specific for a particular gene. Gets around prior probability. So if you have a patient with a well-defined diagnosis of cystic fibrosis, there's only one gene in the world that causes that disease, and there is largely no phenocopies if you have a full-fledged CF diagnosis with sweat chloride testing and the full thing. The prior probability that patient has of pathogenic variant in the CFTR gene is extraordinarily high. So if you just sequence the whole gene and find any variation, the prior probability is actually quite high that variant will be pathogenic. Does not work as well for diseases that are highly genetically heterogeneous, that we don't understand all the causes, that have phenocopies and all of that, like hearing loss, retinitis pigmentosa, diseases that have lots of different genes. But for certain genes where the patient's phenotype is highly specific for that gene, you can use that as supporting evidence. In this case, we largely don't let you use it more than supporting, but for some expert panels where there is this incredibly strong correlation, they may raise it to moderate or something like that. Also, if the variant is found in a patient where you've identified a different cause of disease, that's sort of evidence towards non-pathogenic. So in this particular FBN1 case, the patient phenotype, the reported cases, it's actually interesting in this case. This is not a disorder where the phenotype is so highly specific for this gene that we would count that a lot. And in fact, in this case, the reported cases had such variation in phenotype that sometimes we use that as argument against pathogenicity, because there's more likely a specific variant will likely have a similar phenotype for most disorders. And so when you see the variant in a bunch of different phenotypes, we sometimes start to argue against pathogenicity. In fact, this is true in this gene. And so we said, well, maybe the observations of different phenotypes in this variant were supporting evidence against a pathogenic interpretation. So all in all, after we went through all of those different pieces of evidence, we ended up with 2 pieces of supporting evidence for benign interpretation, maybe at best 1 piece of moderate evidence. That is clearly VUS. Despite the fact that you remember 3 of the labs called it likely path or path. So this is, an example of when you're not looking really thoughtfully at this type of evidence, you can easily go awry and misinterpret a variant. I will say, after that whole group, this is the Caesar submission to ClinVar for this variant. So we went through that effort. We all agreed in the end it was VUS. We submitted to ClinVar. And now all of the other clinical labs also are all in agreement that this is a VUS. So it sits there in perpetuity. Someday we'll know for sure what it is, but not now. I did want to spend a little bit of time on some of the things that we didn't talk about with that one variant, like functional studies. So there are some functional studies that are very good. I can tell you most functional studies that are out in the literature are not very good. Here's an example of one that we do rely on fairly well, and a lot of these tend to be metabolic diseases where the underlying molecular etiology relates to an enzyme that's very easily testable in a human sample. Draw a blood sample and run an enzyme assay on it. And so for the GLA gene, which causes Fabry disease, which is both a neurosensory and renal phenotype, you can do alpha-galactosidase enzyme testing. It's a clinically orderable test and get a measurable value. It's been a well-validated clinical assay. And so when we have a VUS in the GLA gene, we suggest the physician go order alpha-galactosidase enzyme testing, and they will come back with values, 1.5, 7, and in a male with a pathogenic variant, it should be almost 0. In a female, who's a carrier, we'd expect some level of enzyme based on the wild type copy. So you can use this functional assay as very good evidence to inform variation in that gene. On the other hand, as I mentioned, a lot of other, you read a publication, you've got some figure from one research group that shows some effect, and it's very subjective to decide. You may be able to look at the statistics for that one assay that were reported, maybe there's a p-value, but you have to really think about how well validated that assay was. Did they test definitively known pathogenic variants and show an effect? Did they test definitively known benign variants and show no effect? Did they also make it clear how that biological assay actually relates to the phenotype? So that this assay, very clear, we know the GLA gene leads to this enzyme, we know that is the biological effect in the patients. For a lot of other assays, they're like, oh, it decreases receptor binding. Well, does receptor binding have anything to do with how the phenotype develops and the pathogenic mechanism? If you can't say that for sure, then even if the assay is great at figuring out receptor binding, you can't say that that is evidence for pathogenicity. So really connecting that phenotype to the biological assay is very important. Also, the environment. Did they do it in an in vitro system where it's not clear that recapitulates the biological setting? So the closer the assay is to testing true human environments, the more usable that information is. So as an example, our ClinGen hearing loss work group went through reported assays across different genes, things like electrical coupling assays for the GJB2 gene, which is a gap junction protein, dye transfer assays for SLC26A4, radioisotope and fluorescence assays, COPE localization assays. And you can see in this case, we never came up with anything that met strong evidence for these assays. Based on the validation data, we made decisions whether we would count the pathogenicity as moderate or supporting based on validation of benign and pathogenic. In this particular assay, it never got above supporting. This one, supporting for a negative result, moderate for positive, depending again on how well they validated each result. So that's sort of an example of an expert group going into very specific assays. And in some cases, we listed assays that didn't count as any evidence to give better guidance to the community as to how to count functional evidence. Another critical thing, and this is really particularly critical now that we're screening genomes. Yes, question.
Just one quick question. I don't see any phenotypic effects on a model organism or anything that's done in vivo. I mean, these are in vitro. Did you take into consideration in vivo model systems?
Yeah, and it talks a little bit more about that. I didn't give that as an example, but if there is an animal model where they've where they've put the specific mutation into that model, then absolutely that can be considered strong evidence. Again, depends on how closely that human disease is recapitulated in that animal model. And if it's a fairly good recapitulation, then that can be used if you put the specific variant in for sure. That would be very good evidence, yes. Unfortunately, most of the time, we don't have a model for the individual variants we find. It's just very rare. We often have a model for the gene and perhaps disruption if it's a loss of function. That kind of data is often available, but very rarely the specific missense variant you've come across. So getting back to null variants, I mentioned earlier that if you're trying to interpret null variants, the first thing you have to determine is loss of function and known mechanism of disease for that particular gene. And if the answer is no, then you can't, and even if you prove that that is affecting the gene or the protein and knocking it out, doesn't mean it's pathogenic. Now, for a lot of diseases, the mechanisms are loss of function, either recessive, which is often loss of function, or for some dominant disorders, the mechanism is known to be haploinsufficiency, where loss of function is also the basis. So for those disorders where you've established that fact at the start, the next question is, okay, I come across a frame shift variant or a nonsense variant or a canonical splice site variant in the +1, -1, +1, -2 positions, can I just assume that that's causing a loss of function of my protein? No, you can't. And there's a lot of questions you have to ask. Now, when we've been doing mostly disease testing for like the last many years, again, I told you, if you had a patient with cystic fibrosis and you went in and tested the CFTR gene and found a predicted loss of function, the likelihood it really caused loss of function, extraordinarily high. On the other hand, what I do all the time is I screen healthy patients and put filters on to pull any predicted loss of function variant out of any gene. If you do that, the prior probability any one of those variants is pathogenic is much, much lower. You have to be very careful about interpreting and assuming what looks to be disruptive is. So what are the things you ask? How frequently are null variants? So this gets around constraints or the gene level, stuff we talked about last week. But looking at the specific gene, is the exon alternatively spliced? So it may be a really good loss of function in that exon, but that exon is normally just spliced out and not relevant. And that could be just many different splice versions of the gene function in every tissue, or it could be a tissue-specific effect, and such that if this is a gene for hearing loss, maybe that exon is spliced out when it's transcribed in the cochlea and the inner ear, and only used in a different tissue for which no phenotype is occurring. So understanding alternative splicing is critical to make sure that that apparent disruptive variant is actually disrupting the relevant and biological transcript that you're needing to look at. And that can often be determined if there are other known pathogenic variants in the exon, which proves that exon is required for disease, yes, or function. Yep.
I know, so the ACM categories that we're trying to get to, are pathogenic, and it's just about the variant, not about your case, right?
Right.
But with regard to, say, these null variants, aren't, in some cases, we're accumulating evidence that the variant is specific to dominant or specific to recessive. But the ACMG score then doesn't seem to reflect that? You know what I mean?
So we are determining pathogenicity, and when you arrive at your final pathogenicity, you then have to say this variant is pathogenic for disease X in inheritance model Y. So I would say this variant is pathogenic for cystic fibrosis in a recessive manner, which then says to have a phenotype in my patient, I need two pathogenic variant on both alleles. And so we're saying what's the effect on the one allele when we derive our pathogenicity assessment. But then that third step that I said we're not really doing here, to say, is this explaining disease in my patient? That then has to look at both alleles for recessive disorder and say, did I find a pathogenic variant on this allele and this allele? And so I told you often we call these cases positive, negative, inconclusive. There's two major reasons for inconclusive. One is all you found is a VUS or multiple VUSs. The other is you found a pathogenic variant on one copy of the gene for recessive disorder. And your inconclusivity is because you haven't identified a variant on the other allele. And so you may say, Delta 508 is absolutely pathogenic for cystic fibrosis in a recessive manner, but my patient with CF, I couldn't find a variant on the other allele, so I'm interpreting that case as inconclusive. I can't say that that variant is causing disease until I show, it's a little different for CF because it's like only one gene involved, but hearing loss, and in your case, retinal diseases, a lot of these recessive, huge heterogeneity. We find all of the time a patient is a carrier for pathogenic mutations in genes for the same disorder. And later we find a new gene and discover they're compound heterozygous in a different gene, but they're still a carrier for one pathogenic. And that's where the difference in the interpretation of the case versus the interpretation of pathogenicity. Does that make sense?
It seems like if you have a null allele in a known recessive gene, you'll quickly get to it's pathogenic.
Yep.
Whereas if you have a gain of function variant in a dominant, where you have all this pedigree segregation data, you'll also get the pathogenic. But
Good.
those are, I mean, right, let's say you do. So both cases you end up with pathogenic, but they don't really mean the same thing, right? Because you have to.
Well, they mean the same thing in that you're saying that mutation can cause disease in a certain setting, but in the case of the recessive, it's in the setting of the other allele being affected as well, right? And another example of in dominant disorder, so there are certain pathogenic mutations that have very low penetrance and mild effects. So when I'm interpreting a hypertrophic cardiomyopathy case, if I have a newborn or a child with severe disease, which is quite rare for hypertrophic cardiomyopathy, I have early onset severe disease, and I find a particular variant that I know is associated with a mild phenotype, typically very late, I might say inconclusive, even though I've called the variant pathogenic, because I don't think that variant is explaining that phenotype, and something else must be going on. So again, pathogenicity is, can the variant cause disease, which may require a setting, like variant on the other allele, not has it caused disease in my patient for sure, right? So other questions, is nonsense-mediated decay predicted? So often what happens when you have one of these frameshift nonsense variants upstream in the gene is it actually leads to rapid degradation of the transcript, leading to no functional protein produced. That there's a specific rule that, if it's in the second to last exon, the penultimate exon, at least 50 base pairs up from the end of that exon, if that position, if the stop codon occurs before there, it will largely lead to nonsense-mediated decay. There are exceptions to that. It's not 100% foolproof rule. But we tend to use that because it's largely true to decide whether a variant based on position is likely to cause disease. You can also assess where other null variants have shown up, and if there are null variants that are downstream of your variant, more likely that it is going to lead to an effect. Whereas if you just found something that's the most 3', be very careful, especially if it's after that nonsense-mediated decay site. So those are all things that you have to keep in mind when you're interpreting what looks to be a frameshift nonsense canonical splice variant that's likely to disrupt and whether it really might disrupt. And here's an example of a variant where you look at all of the transcripts and can say, yes, this exon is present in all known transcripts, that's good. But then here's a variant that we found in a babyseat case where there were 2 biotinidase deficiency variants. One of them, this one, was a very well-established, well-published, go through the ACMG guideline, and it's pathogenic. But we detected this novel +1 splice variant in the first exon. So you're like, well, it's upstream of nonsense-mediated decay site, that's good. But if you look at this particular exon, there are actually 2 putative translation initiation sites in this gene, one in exon 1 and one in exon 2. And there's never been any pathogenic variants reported in exon 1 in the literature. And if you look at GTEx data, actually this just shows the different transcripts. That exon is present in NCBI and RefSeq gene in 3 out of 5 transcripts, but it's absent in 2 out of 5. And then you look at GTEx data, and you can see for all these different tissues, this exon 1 is only present in the testis. So immediately you go, I'll bet you this exon is not required, and I need to interpret this variant as uncertain significance. So that's an example of being very careful about assuming the effect of what looks to be a canonical splice site variant, because it may disrupt this, but based on this data, losing that may not be a big deal, particularly for given that it's only expressed in testis. Now, interesting in this case, because this was a newborn who had newborn screening results, and newborn screening includes biotinidase deficiency testing, we asked to get access to the results of that biotinidase assay in the newborn. And it turns out that baby failed newborn screening just for biotinidase deficiency, but then what they normally do is they do a repeat screen, and the baby squeaked by, and they sent the baby off with a normal report. Nobody ever knew that this happened. But when we got this result, we're a little suspicious about it, and then saw this, it's a little suspicious to happen to see this failure on newborn screening for just that one result. So we decided it was worth calling this baby in, getting an actual enzyme level testing, and in fact, the baby had an abnormal reduced enzyme level. And that baby has now been put on biotin and is being followed for potential risk for biotinase deficiency. Probably what they have based on their enzyme level is partial biotinase deficiency. And in fact, if you go back to the biology, first of all, these are all adult tissue data, right, in GTEx. So keep in mind, this does not represent the fetal state nor the childhood state. It doesn't test every tissue also. And so, it is very possible that is having an effect and reducing the level of viable transcript, which may, be important, but it's a partial effect, which may correlate with why they kind of are having slightly failed, not failed results. Stephen's been making this figure, which is a flow chart to make decisions on how to interpret loss of function variants and do they really cause loss of function, asking questions about is it predicted to undergo nonsense-mediated decay? If it's deleting an exon, is that exon take the protein out of frame or in frame? So a whole series of questions to then arrive at different strengths of evidence modifying the ACMG code PVS1 from strong, very strong, moderate to supporting. So we're trying to give the community a little better guidance for what exact questions to ask and then how to end up with an answer of evidence based on what you got. So I'm not going to walk through because we're running low on time. Also, for those of you who want to curate variants, don't have your own curation interface, and want to use the ACMG guidelines, you can go to our curation.clinicalgenome.org. By the way, if anybody wants a copy of these slides or the ones I showed last week, just let me know. I'll probably just post them on a Google link. But you can go here. You have to register and get a username account, but anybody can do that. And then you can go in and use our variant curation interface. And you'll see all of the codes that whether they've been applied or they're shaded in. And so you can go through, look at the codes, and add what strength of each code, put them all together. Once you've done that across the codes, it will then give you a summary of everything you've applied, whether you modified the strength, any notes you want to put in for why you applied that code, what the criteria you said were not met. You don't have to check them all, but whatever you are contributing to the criteria, you select. And then it will give you a calculated pathogenicity based on the codes you applied. You can, with your professional judgment, decide to modify that and say, well, it says pathogenic, but if I look at the aggregate of evidence, not quite sure it's quite there, I'm going to modify it to likely pathogenic. You can do that, but you have to give a reason why you did that. You associate with disease and a mode of inheritance. Remember, I said that was critical. And you save it. And so that's what our expert panels are doing, and anybody can go in and do this for a variant if you want to track it. We will also help you submit it to ClinVar. We're getting that functionality set up. So I'm going to, I had a few other examples and other rules, but in the interest of time, I'm going to stop there, open up for any further questions that anybody might have. Thank you.